All right, today we are talking about section 3. We're skipping section 2. Section 2 about section 2 will not be on the quiz or the unit test. Sections 1 and 3 are the most important. So, advances in genetics. And what we're talking about here, most importantly, is selective breeding, because that's going to be a tie over to what we talk about next week. These other questions we'll go over briefly, but that's the most important one. So let's talk about the two types of selective breeding. Who can tell me about one of these or two of these? Okay, so that's exactly correct. Inbreeding is breeding two organisms with similar characteristics to get the desired traits in the offspring. Now, that sounds, again, but who can think of an example about this? Because, of course, we do this. This is how scientists, what's an example of inbreeding that you might use? Yeah. So dogs, and we'll watch the dog video probably tomorrow as we switch into talking about natural selection, which is a little bit similar. But in dog species, you can breed. We breed things like you've got German Shepherd dogs, or Dobermans, or hunting dogs, border collies, dogs that are bred for very specific purposes. And humans have been inbreeding dogs for thousands of years, right? To get dogs to do what we want them to do. We've bred dogs to be cute and tiny and look like puppies their whole lives and be small enough to sit on your lap. Or we breed dogs to do jobs for us. So, and in case of the dog species, you're breeding similar ones, and you are sometimes also breeding related animals, which we'd never do in humans, but we do in animals sometimes. Does anyone know any issues with inbreeding problems you might have? Yes. So the good thing is you get these dogs that are bred to do specific jobs, and they're very good at it. But the bad thing is if you are breeding similar animals, related animals, if there are recessive genes, bad genes that are in that gene pool, they're going to start showing up much more often because you've just started breeding very similar things together, very similar dogs together, even litter mates or cousins or whatever. So you breed them together, and then those recessive traits might show up, and they might be bad things. We know that purebred dogs, especially some of the big dogs like German Shepherds, they have hip dysplasia. Or some of them might have, everybody knows some dogs that are a little too inbred that maybe aren't very smart, right? Some of the little bitty dogs you'll see, their jaws, they have like an underbite, right? Which, if you're just eating dog food, if your dog's just eating dog food and you're taking care of him, doesn't matter. But some of these dogs would never survive in the wild because their teeth don't line up quite right. They would never be able to survive. Sometime, and okay, so let me see, a toy dog or a lap dog. So, and then another example, the fast plants that unfortunately we did not have very good luck with this year, but we read about that. That was through selective breeding, through inbreeding. The scientists were able to keep breeding for a smaller and smaller plant, for a plant that would grow with a short life cycle. And so inbreeding can be very, very useful. And maybe in some ways more useful in something like plants than animals. We certainly do use it a lot, but you get some bad things. In plants you do sometimes, but it's not as big of a deal, right? Your plant doesn't have to be smart. Okay, so then what is hybridization? What is hybridization? Yes. Okay, so in hybridization, again, it's going to be kind of the opposite of inbreeding, crossing organisms with very different traits to get new combinations. A good example of this is if you have a dog who's a mutt, and a lot of times when you get a dog that's a mutt, they'll say, oh, sometimes they'll actually know. I had a dog for years who was a black lab border collie. And he could be like totally easygoing. His fur would dry really quickly. He had webbed feet. He loved the water, but he was also really smart and could be a little bit high strung at times too, which is kind of weird to be kind of easygoing and snuggly and kind of high strung. So sometimes you'll know the two traits. And in that case, it was a nice combination of the two very different things. But the problem you can imagine with hybridization is you don't know which traits you're going to get. You might get the traits you don't want so much from both of them. It's a little bit unpredictable. But with hybrids, we get something that's called hybrid vigor. Vigor just means you're strong. So hybrid vigor is the opposite of the fact that these dogs, the purebred dogs, might have problems because you've got that inbreeding. You've got traits that show up. You've got recessive genes that show up. Hybrids, it's the opposite. You've got that strength of lots of different genetics, and so they'd be able to resist disease, or they generally might be healthier or more mild-mannered, or that sort of thing. Okay, so those are the two main types of selective breeding. And again, when we look back at the word selective breeding, it just simply means that we are choosing which traits to should be included. So selective breeding is choosing which traits get passed on. And again, as Tim and Moby mentioned in the brain pop we watched earlier, when you talk about cloning, cloning is just making an exact copy, either usually by taking a cutting of something. And people have been doing that for, as long as people have been farming and planting things, because you want to plant the ones that grow well or that produce lots of seeds or fruit or vegetables. So the next subject that we'll talk about next week is actually natural selection, which is nature selects which traits are best to survive. So whatever traits are helpful get selected by nature to be passed on. Oh, and earlier, actually, we'll come back to that. Okay, so these other things real quickly. Cloning, we talked about cloning, making an exact copy. It can be as simple as taking a cutting from a plant, or as complicated as tricking a cell into thinking that it's a fertilized egg and growing into a new living thing. And so we talked a little bit about that. Bacteria, real quickly, and this, these things are kind of extra. How are bacteria used in genetic engineering? Did you guys read that? Bacteria and viruses. Bacteria, so who's got the answer to that? How do we use bacteria? All right, so just zooming through these, bacteria, short life cycle, and because of that, we can get them, you can actually use a virus or something like that to put something in the bacteria's DNA, and then the DNA will reproduce it, so you can get it to make molecules. Gene therapy would be what if somebody had a genetic disorder, if you could go in and fix it, like before the baby was born. Human Genome Project was a genetic mapping project, I believe in the 80s and 90s, and mapping the human genome. So looking at all of those genes and looking at all those chromosomes and saying which genes are on which chromosomes. And that's going to tie in a little bit to the video we're going to watch here in a few minutes. It's been very useful because if you know which genes cause which diseases, things like breast cancer or other things like that, you can actually, when they treat people for these diseases, they give them a treatment that is specifically tailored to that person. So instead of just broad radiation that makes you sick, it can be a more specific treatment. Okay, so what we're going to do next is we're going to watch a little bit of a NOVA video. We won't watch the whole thing. We'll just watch about 15-20 minutes of it. And this is about what if you could find out what your genetics are. So would you want to know your genetics, your entire genome? Would you want to know if you have genes that might mean you get sick someday? Would you want to know so that you could prepare? Or would you rather just take what life gives you? So anyways, we're going to take a look at that. Quiz tomorrow over sections 1 and 3.